Now we will move on to this chromatin epigenetics or chromatin structure based epigen. So because you can imagine the direct methylation of the DNA definitely influences the expression, because at the end of the day, what does it do? The epigenetic modifications, it bring the changes in the expressions, right? Either activation or repressions, it happened at a DNA level. But here it's more complicated because the modifications happening on the protein level, histone proteins, and then this protein regulates or decides the associated DNA to be switched on or switched off. And that is why these protein modification is considered, although not on DNA or RNA, the modification on the protein, it's considered as significantly as an important epigenetic modification, because it directly influences an associated DNA, right? So very important epigenetic modifications. In eukaryotes, I said these histones as highly useful are essential to compact the DNA. So not only for compactions, it also for regulating the expression. That means the compaction and gene regulation, it works together. So that is needed. Who does it? We know broadly it's a chromatin remodeling factor. That's what you might have read in many textbooks. But beyond that, at minute, at molecular level, the epigenetic modifications play a very important role in the chromatin structure based gene expression. That means chromatin packing based gene regulations. So before I start explaining, where I will explain in detail the important epigenetic modifications on histone proteins, I'll walk through or explain in a basics in a couple of classes how these chromatin structure is arranged, packed, what is briefly about remodeling and so on, so that you will understand these basics. When we go for a modification based, we'll connect this knowledge in a chromatin based gene expressions, right? So this is how our DNA is packed in the nucleus. So if you take out these 23 pairs of chromosome each and every cell, if you put it on a table, as I said in the introductory class, it will be roughly 2 meter plus. So such a lengthy DNA polymer. So these DNA need to be carefully arranged, packed systematically, very important process, and packed within the size of nucleus 5 to 10, depends on cell to cell. Some cell will have even 20 microns. So but it has to be packed into tiny nucleus. That means the packaging is very important. So the packaging is happen with the help of histone proteins, and now we understood with the help of epigenetic modification. Obviously, the chromatin remodeling machinery also utilizes the epigenetic modification, because that also it does the chromatin's nucleosome sliding or movement, whatever, with the guidance of histone modification. It doesn't do independently. So that means there is a strong connection between the chromatin remodeling versus epigenetic modifications and packing and gene expression and so on. Okay, so for your informations, chromatin is a nucleoprotein complex. The DNA and protein ratio. So in this case, a protein which includes histone and protein which are associated with the nucleosome. Some protein will be just it's a kind of nuclear protein which bind transcription factors, or chromatin bound protein remodeling, which include all the proteins, and it is roughly 2:1 mass ratio. Whereas histone and protein DNA, if you take it directly, 1:1 ratio. So this is how it exists in a cell. And there are several layers of condensations, a level of condensations. The initial is 10 nanometer fiber condensation of the DNA. That is roughly it's a 7-fold compactions. So this is you can consider as a kind of euchromatin structures, or active chromatin actually. So that means this anyway DNA is not free in a cell nucleus. So the lowest level of compaction is 7-fold. So this fold is readily accessible for a event transcription factors, where it can bind and mediate a gene expressions. But obviously, while doing it has to slide the nucleosome, I mean histone protein, because then only these polymerase can move on, right? The next level of compaction is 30 nanometer. So that's a condensed form, inaccessible to the even transcription factor and polymerase. So this is the important change from euchromatin to heterochromatin. This is euchromatin to heterochromatin. The changes from the 10 nanometer fiber to 30 nanometer fiber. Then further it makes more condensed to as a compact chromatin, then chromosomes structures. So this is how the order goes on. So as you all know, the nucleosome is a fundamental unit of chromatin structures. So the nucleosomes defined as a DNA protein complex, the single molecule, I mean the single structure, you call it as a nucleosomes. So basically the DNA wrapped on the histone octamer, 8 proteins, 4 protein, but they all dimer and octamer, and then this unit you call it as a nucleosomes. Right. This is a schematic picture of these heterochromatin versus euchromatin. You see the heterochromatin, it is a transcriptionally silent. So that's why I explained this part. So when it goes to compaction level, it is a completely silent, that is inaccessible for either transcription factor or polymerase for sure. And whereas euchromatin, it is a transcription active. That you can see when you take a normal cell, when you culture the cells, when you do with the simple DNA staining, for example DAPI or OSHA, you will see the intense DNA spot stain. Like in the nucleus, you will have very dark blue color stain. This is basically heterochromatic structures. So condensed DNA, where more DNA molecule compacted, that's why the more DNA staining is present. And but then also you will find very loosely stained blue color structure. This is, you can consider or call it as a under the euchromatin. That's what marked here. It's not very clear. This is heterochromatin, you can see here, and this is euchromatin. These are all roughly in the blue color structures only. And more importantly, there is a strong coordination between these structure formations and then nuclear membrane, because these heterochromatin tend to anchor onto the almost nuclear periphery. So you have 23 pairs of chromosomes. Each chromosome will have the euchromatin, heterochromatin. It's not that chromosome specific. That means all heterochromatic regions of all the chromosomes, right, need to be assembled together to form a structures. So that will be achieved now by anchoring under the nuclear membrane or nuclear periphery. In fact, now this is even defined as a kind of phase separation of this heterochromatin. This is a new term used, the protein molecule which undergoes for these membraneless structures in the cells. So these are called the phase separations. So heterochromatin, it's a classical example of phase separations, because all the condensed structures from the various chromosomes assemble as a repressed chromatin. But just to have a brief comparisons between the prokaryotes versus eukaryotic gene expressions for your just refresh. In prokaryotes, the major is, you might have heard about it or come across, promoter, operator, and then regulatory elements. But the key difference is the prokaryotes mostly non-restrictive, because it's a free DNA elements, does not have any chromatin like structures, whereas the eukaryotes ground state always restrictive. So you need to have an assistant there, you need to have a chromatin remodeler or epigenetic modifier for a successful opening or removing of these histone proteins to have a active transcription. But in this case, prokaryote is very different. You need to have an operator and then regulatory sequences, and then activator can go and bind directly and activates. So no need to have much of complex process. But in eukaryotes, very complex, as you understand. It is tightly packed on the nucleosomes. Yes, in a eukaryotes, you call gene activations. That means either you call it's anti-repressions. That means you remove the repressor molecule which is bound. So what are the repressor molecule can bound and make it more heterochromatin? almost all the repressive epigenetic modification can recruit the repressor molecule, and that makes more condensed state. That means you have to remove that repressor molecule. So that will be done with the help of chromatin remodeler along with the transcription factors and so on, and histone chaperone. So basically, anti-repression defines in eukaryotes, you remove the repressor molecules with the help of other molecules. Other molecules can be transcription factors or histone chaperone or even chromatin remodeler. And other way of activation, after this you recruit directly the polymerase, which takes care of the transcription. So this is how it goes sequentially. So in eukaryotes. So how does these the chromatin remodeling takes place? With LFOP. Of course, I'll just show you in detail in upcoming classes. Just for your refreshing purpose, the histone methylation or histone acetylations, which it takes very important part in chromatin remodeling, and particularly acetyltransferase or methyltransferase enzyme, which contributes nucleosome remodeling by extensively modifying the histone tails, because this is the triggering factors essential for the remodeling. Remodeling is essential for your normal transcription or gene activations or conversion of heterochromatin to euchromatin, same from euchromatin to the heterochromatin. In all the cases, remodeling is very important, where the underlying player is epigenetic modifications, and that's done by acetyltransferase or methyltransferases. The second, it's through help of specific complexes, which is the ATP dependent process, which I'll highlight very briefly. And what happens during these process, the remodeling process, which creates a space for the transcription factor and polymerase, and then which accessible, and then which lead to the gene activations. So this is how sequentially it happens, right? So very briefly, the tail I said, the stone tail in the nucleosomes, right, just projected out, which is shown here, play very important role in the remodeling and gene activations. How? This is the place where you have extensive epigenetic modifications. So there is a two at least theory as well as experimental evidences. The histone protein is basic proteins, already as said, right? And then nucleic acids, DNA is a negatively charged. So because of this charge, it has a strong attraction and then binds very strongly. But in this case, the acetylations is always a gene activation mark, or in a chromatin opening, for example, acetylation play very important role. If you want to convert the 30 nanometer fiber or heterochromatin to euchromatin, or a cell want to express temporarily euchromatin genes, the first steps, what does it do? It introduces the acetylation mark. Acetylation is a gene activation mark. So why acetylation first? Because acetylation brings negative charge, and when acetyl group added and there is a charge imbalance, which results into the repulsion between these, or at least opening of DNA from these stones little bit, which allows for the transcription factor and the remodeling complexes goes and bind and remodel, which makes for the polymerase to go and bind to this mRNA synthesis. So basically, the acetylation on the histone protein brings the charge imbalance. That's a very first steps happens. In addition, acetylation also play very important role in recruiting the activator complex. That is second. The first thing is biochemically what happens, this brings a charge imbalance because it converts lysine, which is already positive charge, and because of adding to the acetyl group here, it neutralizes charges, and which obviously loosens the DNA histone interactions. So this is what biochemically happens in the cells. So because I'll show you in specific examples how these acetylations play very important role in the conversion of 10 nanometer to 30 nanometer fiber. So keep in mind, in two places I highlight this process. Some of the lower eukaryotes, this one, yeah, between these two, uh, 10 to 30. Some of the lower eukaryotes does not have a H1 proteins. I said H1 protein is essential to form a higher order chromatin structures. We have two nucleosomes like this, where H1 will binds here and helps to condense the nucleosomes. In addition, the acetylation also play very important role. Basically, reverse role. Removal of acetylation favors these condensations. So only these two events are essential. Convert 10 nanometer to 30 nanometer fiber. Okay, so with this I'll just stop it. So the point here, the acetylations play very important role in opening the chromatin structure in the beginning. The first, how does it do? It brings a charge imbalance. It neutralizes charge, which helps to weakening the DNA histone interactions, which promotes more and more nucleosome remodeling complex, which binds and then which opens, allows for the transcriptions. So this is how it happens in chromatin packing. How it bring the charge imbalances, because the histone proteins are highly basics, and nucleic acids DNA negative charge. Because of the charge, it has high possibility or through high chances of ionic interactions, which makes more DNA protein interaction in this case. And acetylation brings the or nullify the positively charge of the histone proteins, which weakens the histone DNA interaction, which favors for the opening. This is what I stopped. Let's walk through the how the DNA is organized in the eukaryotes, which I showed in the earlier classes as well. You have the free DNA molecule, right? The first level of compaction, it has to wrap on the histone proteins. This is the histone proteins, major 4 histone proteins, which I'll show you. This folding is roughly 146 base pairs, roughly. This is 1.7 fold wrap. So on a single histone core protein. So this is nucleosome structures, histone core wrapped with the DNA, and then you have the linker DNA, and then again, so this is how this compaction goes on. I already explained the methylation at the various levels. Yes, so this is 146 base pair of DNA wrapped on a single nucleosomes, and this is the A from atomic force microscopy structure, which clearly shows the nucleosomes looks like beads on the string like structures. String. So this is typically like beads string structures, A from microscopy structure, which shows that how DNA is wrapped on the histone proteins. This atomic resolution level of a single nucleosome, you can see that DNA is, yes, wrapped on the histone core. This is histones, the 4 histones, H2A alpha, H2B beta, and H3 and histone 4, and each exists as a dimer, basically H2A, H2B, H3 and H4, totally 8 proteins. So this is an histone octamers. The unique feature I already explained, the tail, the end terminal tails of each proteins. It has an end terminal tail which projects out of the nucleosome core structures, and this is the place the most of the epigenetic modification takes place, which it decides packing, gene expression, unpacking, and so on. So these all things I briefly introduced during the introductory classes. Okay, let's move on how the real packing happens in the cell, eukaryotic cells. So one of the classical biochemistry assays in the initial days to understand how the nucleosomes packed in the cells, they use the simple biochemistry assays. So the biochemistry assays, it says you need to isolate the chromatin from the nucleus, It's called chromatin isolations. And then when they subject this chromatin to DNAs, and particularly MNAs, it's a very important enzyme, micrococcal nuclease. So the nuclease DNase enzyme, it's an endonuclease enzyme isolated from these bacteria, micrococcal, highly efficient in most of these DNA structures, and particularly it is used extensively to prepare the mononucleosomes. When after chromatin isolation, when it's subjected to the DNAs, it has a preferential cleavage only on the linker or free DNA. Free DNA means this DNA devoid of histone proteins. Of course, the DNA wrapped on the histone protein little bit resistant for the DNA's digestions. So after that, you will have this nucleosome core particle. So this is how in early days they isolate the nucleosome core particles. So which it tells that yes, the DNA is packed beautifully on the histone proteins, and the DNA wrapped on the histone proteins are resistant to the DNA's digestions. And then later, to even to separate one of the simple biochemistry you can always think about it, since the histone DNA interactions are charge dependent ionic interactions, when they increase the salt concentrations beyond physiological level, obviously it tend to dissociate, right? So it breaks the ionic interaction when you increase salt concentration. It can be potassium chloride, sodium chloride. So normal physiological concentration roughly 100 to 150 millimolar. When you make a buffer towards 250 millimolar, obviously all your DNA histone interaction will be broken down. Then you have separate DNA molecules and histone octamer. So this is how they identified, yes, there is a 146 base pair wrapped on the after this separation, they run a gel and then they see where it mobilizes. They found yes, it runs. These are all before the coming up all the NGS technique. And then further the dissociation of these proteins identified the size of these proteins. So this is how the chromatin structures was elucidated. So one of the common features about these histone proteins, I said histone is a very important proteins, mostly present in the eukaryotes. Bacteria does not have. The archaea as a similar to the histone protein, not histone proteins, which is does some function like again chromatin packing in the archaea. But in eukaryotes, all the eukaryotes, you take from the single cell amoeba or yeast, from such a lower microorganism to multicellular organisms, histone contributes for the chromatin functions. Now the key feature, if you look at it, this is the only proteins, histone proteins, highly conserved across the species. You see, it took really 500 million years for a single amino acid changes. So most of the protein also across, when from compared to the lower eukaryotes to the human, there are a lot of sequence changes, adaptations as per the organism requirements. But histone is the only proteins highly conserved. That's one of the very unique features about the eukaryotic proteins. Across all proteins, this is the one of the proteins which it does not get evolved much, or it has highly conserved in nature. The second important feature about these proteins, most of the eukaryotic mRNA undergoes to post transcriptional modification, particularly poly-A tailing. You might have heard about it, right? So mRNA at the end of the 3' end, it undergoes for the poly-A tailing. This is important event for the transcriptional regulations, stabilization of the mRNA, or in improving a translation of the eukaryotes. The poly-A addition is important for mRNA. Histones are the only mRNA, or histone coding mRNA does not undergo for the poly-A tailing. So histones mRNA devoid of poly-A tailing. Okay. So these are the two important, these are the RNA level important features unique among the eukaryotic RNA, and at protein level across the eukaryotes, this does not evolve much, not much of mutations, highly conserved. That's the beauty about the histone proteins. Right. How does it? A lot of experiments have attempted to show how this assembly takes place. Because assembly of the histone core is very important for a successful nucleosome structure formation. Because you have to think about it at all the level. Because DNA need to be wrapped, yes, and the wrapping should be proper for a successful chromatin formation. If there is any issue in the wrapping also, you have the chromosomal aberrations. So for that, the nucleosome core particle formation is highly essential. So how does it assemble? These are all has been shown using various biochemical assays. The first histone 3 forms a dimer, strong dimer, homodimer structures. And further, this histone 3 homodimer forms a complex with a H4 homodimer to form a heterotetramer. This is parallel assembles, right? Similarly, H2A, H2B also assembles. Then later, all proteins forms as a histone core that you can see here. So you have N-terminal tails, and then this is a histone core. So this is how, at least the biochemical evidences confirms the histone proteins are assembled to form a single nucleosome structures. Yes, so at least the evolutionary analysis shows that the histone 3 and histone 4 even highly conserved compared to the H2A, H2B. Even H2A, H2B are even more of variants are reported, and few additional mutations are reported. But whereas the H3, H4 is kind of highly conserved across out of 4 proteins. So another important feature about this nucleosome structure. So this is how DNA is wrapped. And you all know about the two important features about this DNA structures. Minor groove, these regions, right? This is a major groove. And the minor groove of the DNA structure, double helical structure, is an important contact point to the histones. So if you look at it, the minor groove is the one which mediates the histone DNA interaction on the nucleosome structures. The more importantly, again the biochemical evidences have shown that the minor groove having these HT rich regions highly preferred to interact with the histone proteins. So basically, the successful nucleosome structures, the DNA should have a not always, keep in mind, okay, this is a preferential, but the GC rich minor groove also may bind to or will bind with the help of assembly factor to the histone protein and form a complex. It has to form, because there are a lot of organisms as a GC rich, and then how does it forms the nucleosomes? Yes, it does. But these biochemical evidences comes compared to the GC rich minor groove, but AT rich minor groove prefers, or TA rich minor groove prefers the stronger interaction. That's it. But that doesn't mean it's always in the cell, it always prefers only AT. So this is only for informations. Yes, when you do the comparative study of these nucleosome structures which are having the AT or TA or AA compared to GC, it has a better affinity. It is an in vitro studies. Yes, the in vitro studies confirms that across the organisms, there is synthetic one or chicken stones, or yeast, in all the places, different DNA structures, people have studied the AATAT minor groove favors for the stronger affinity with the nucleosome core particle. Right. So another features about these nucleosome structures in connection with the genes, particularly transcriptional start site. TSS is a transcriptional start site which is near to the promoter regions. You can see here, this is a promoter. Again, most of the cases it has been identified, the TSS are having a less or minimal nucleosomes. This maybe highly active genes or housekeeping genes. This makes sense. So that means it's a free for transcription factor binding and keep initiating the gene expressions. But again, the transcription factor binding to the TSS and all this depends on the promoter activity, right? The promoter is a very key regions which determines whether the transcription factor to be bound and then initiated transcription or not. So, it depends on the epigenetic status of this promoter, the function of the transcription start site will be decided. What is the information you have to keep in mind? TSS are having the less of nucleosomes, most, not always again. Okay. And another important feature, the TSS having the rich of poly A and then poly T sequences. Again, now you can ask question yourself, because the nucleosome structure formation preferred with the TA, right? Here the TSS mostly has the poly, so why it's not forming the nucleosomes? So this is what they have evolved. So the nucleosome structure formation preferred with the TA or AA sequences, but when you look at a DNA sequence, most of the DNA sequences, again I am not telling it's a global, on a specific, definitely there is a variations. In a general, it has the most of the poly A or poly T sequences in the TSS at transcriptional start sites. So these are the some important features about the nucleosome structure and its presence and so on.